Hello and welcome to the January RIV call on disease-modifying therapies for type 1 diabetes. Today's presenters are JDRF Scientists Cassandra Bazile, Ph.D. and Jay Tinklepaugh Ph.D.
Hi, everyone. My name is Cassandra Bazile, and I'm very excited to talk to you all today about the work happening in the disease-modifying therapies pipeline. But before I talk to you about the research, I wanted to remind you of JDRF's many research priorities. JDRF has a mission of improving lives of people with T1D by accelerating breakthroughs that cure, prevent, and treat T1D and its complications. In a nutshell, our research priorities are focused on two core programs: Cures and Improving Lives, with the Cures program consisting of 3 research areas, which are global universal screening, disease-modifying therapies, and cell therapies. Another critical piece of our strategy we pride ourselves on is the training of researchers and clinicians committed to T1D research. Focusing in on disease-modifying therapies. Disease-modifying therapies are therapies that can change the course of disease. These therapies are aimed at every stage of T1D, such as stage 1, where you have normal blood sugar and start to express autoantibodies, all the way to stage 4, where you have established T1D. Autoantibodies developed before or during stage 1 of disease are used to screen for T1D, and screening is extremely important for the diagnosis of type 1 diabetes. The overall goal of the DMT project area is to prevent, halt, or reverse the loss of beta cells. And this can be done by using therapies towards the immune system, beta cells, or both. The focus of the disease-modifying therapies research strategy is twofold. One goal is to turn off the autoimmune attack against beta cells, which I'll be covering today. And the second goal is to create and sustain beta cells, which my colleague Jay Tinklepaugh will be discussing today. The ultimate goal is to develop therapies that both control autoimmunity and preserve or grow beta cells, and JDRF is playing an active role in accelerating the development of these therapies into clinical testing and eventual regulatory approval and access. For the first part of this presentation, I'll focus on what is being done for the immune side of the portfolio, where we are focused on determining how to turn off the autoimmune attack against insulin-producing beta cells. JDRF has put a lot of resources into understanding the immune system and its role in T1D. The primary function of the immune system is to determine what is yourself and is not yourself. And a breakdown in the ability of the immune system to determine self from non-self can lead to autoimmune disease, such as T1D. It's important when creating therapies that we maintain a balance so that the immune system provides enough surveillance and recognition to identify and eliminate threats, but refrains from being so active that it starts to recognize and attack your own cells as a threat. In the stages of T1D, autoimmunity happens long before stage 1 and is confirmed by the presence of autoantibodies to self-tissue. This means that the turning on of the immune system to your own cells happens prior to stage 1. Therefore, it's important to not only understand what causes activation of the immune system, but also develop therapies that can stop immune activation, as well as therapies that treat every stage of disease. When we talk about immune activation, what we really mean is antigen recognition. An antigen is any particle that can activate the immune system. We generally think of this as bacteria or viruses that need to be eliminated, but it can also be toxins or proteins. The immune system has cells that can present these antigens called antigen-presenting cells, and what they do is identify these antigens and present them to T cells. T cells recognize antigens through the binding of the antigen receptor called the T cell receptor. And the antigen a T cell can recognize is determined by the specificity of the T cell receptor, and this is termed antigen specificity, which I'll be referring to frequently today. Once a T cell recognizes an antigen, the T cell then becomes activated where the T cell can proliferate, cause inflammation, and infiltrate the tissue where the antigen comes from. When the antigen being recognized by the immune system comes from cells in your own body, this is termed an autoantigen. In terms of T1D, the recognition of autoantigens from beta cells initiate an immune response, damaging the pancreas, leading to disease. For the purpose of today's talk, we'll mainly be talking about T cells and their antigen specificity and how we're harnessing the information we get from T cells to create therapies. Our understanding of the immune system has recently led to a huge breakthrough for T1D. After decades of research and funding from JDRF, in November last year, Tzield, known as teplizumab, was the first approved disease-modifying therapy to treat T1D. Tzield works by targeting and altering T cells, and Tzield is approved for people with stage 2 of T1D and delays progression of onset for an average of 2 years. There is currently another study underway called the PROTECT trial that will test Tzield and its ability to slow down the loss of beta cells and preserve beta cell function in children and adolescents with stage 3 of type 1 diabetes. This study will be completed in May of 2023. We've made many clinical advances that use the immune system, that target the immune system to treat T1D. The immune strategy to developing disease-modifying therapies for T1D consists of 3 primary areas, which is to stop the autoimmune attack, to enhance regulation, and to disrupt inflammation. Clinical progress has been made in each of these areas. For stopping the autoimmune attack, the approval of teplizumab, as well as the testing of several other agents, have shown significant progress. For enhanced regulation, approaches such as bolstering Treg function, in addition to transferring Tregs, have been clinically proven to be safe. However, there still needs to be work done on efficacy. And finally, efforts that disrupt inflammation, such as using agents geared at cytokines, have showed promising clinical results. Recently, there have been several advances in technology that have provided us the ability to develop therapies that can further enhance the efficacy of drugs aimed at stopping the autoimmune attack and disrupting inflammation. One way JDRF is doing this is by funding research into the development of targeted immune therapies. While we have recent success with therapies targeting components of the immune system, we may need to get more specific with the types of cells we target for safety and efficacy. As it pertains to on both sides of the disease-modifying therapies portfolio, we can use targeted therapies for immune cells to identify antigens and receptors that cause disease, so we can target just the immune cells that drive disease to avoid broad immune suppression. We can also use targeted therapy to sustain beta cells, so we can deliver regenerative or proliferative therapies, especially to beta cells, avoiding off-target effects. Together, the goal of both of these approaches is to develop safer and more effective therapies that can be used to stop the autoimmune attack, promote beta cell health and survival, or ideally both in combination. Today, I will discuss work being done using targeted therapies to stop the immune attack. So T1D is caused by T cell mediated destruction of the insulin-producing beta cells found in the pancreas. And although other types of cells have been shown to have a role in T1D, therapies that disable T cells have been shown to be extremely effective in preventing and treating T1D. We know that T cells express a T cell receptor, and these T cell receptors recognize molecules termed antigens. And depending on the antigen specificity of T cells, they can cause autoimmune disease. Current immune therapies utilize broad immunosuppression, which means the therapy targets a broad population of cells, no matter the specificity. Broad immunosuppression is represented in this cartoon, where I show T cells with different antigen specificity, with the red T cells recognizing self-antigen, causing them to be autoreactive. In this example, broad immunosuppression leads to the deletion of all T cells, no matter their specificity, including T cells that are not involved in autoimmune disease. In contrast, antigen-specific therapy only targets the cells that have been shown to cause disease. Here again, I show the same figure with the red T cells being autoreactive. In the case of an antigen-specific therapy, the therapy would only target the autoreactive T cells, leading to depletion of only the autoreactive cells and allowing the non-autoreactive cells to survive. In T1D, antigen-specific therapies can precisely target and disable the immune cells and are promising for preventing and reversing disease. However, for antigen-specific therapies to work, we must first understand the specificity of T cells driving T1D, and secondly, we must be able to determine the antigens that T cells are targeting. JDRF is currently funding several investigators that are using unique technologies to identify autoreactive T cells, as well as their antigens, so that they can be used to create therapies. I will now discuss some of the projects that are ongoing in this area. Dr. Maki Nakayama at the University of Colorado is pursuing the development of a technology that determines the antigen specificity of T cells from people with T1D across all stages. Her lab has established a screening system that takes T cells from blood, islets, and lymph nodes of donors and combines them with newly identified antigens from the pancreas. Upon activation of these T cells with islet antigen, the T cells emit a light, allowing for her lab to identify T cells responding specifically to islet antigen. She will use this platform to test 80 antigens, allowing us to gain significant insight to the types of T cells that initiate disease, as well as providing potential ways to target these T cells using therapies. Another lab that is working to identify the specificity of T cells in T1D is the lab of Dr. Michael Betts at the University of Pennsylvania. The Betts lab has developed a strategy to study the direct interaction between T cells and islets from the same donor. This work is especially unique since for the first time, he will reproduce a system that happens in humans in an in vitro system. The Betts lab will isolate T cells and islets from the same donor, and through a co-culturing system, will be able to identify which T cells respond to antigens from islets. This work will allow us to understand how autoreactive T cells interact with islet cells to cause their destruction and potentially reveal novel pathways and mechanisms that can be targeted for drug development. Lastly, Dr. Stuart Mannering from the St. Vincent Institute in Australia is focusing on discovering new antigens. The Mannering lab was previously part of a collaborative group that identified a new class of antigens that T cells respond to. The antigens that his group discovered come from stress beta cells, and they're able to activate T cells in people that have T1D, making these T cells likely to play a role in disease. The Mannering lab is now working to identify new antigens utilizing a system that uses yeast to express islet antigen. The identification of antigens are essential for the development of assays to not only screen for autoimmune disease, but to create antigen-specific therapies to prevent or reverse T1D. To summarize, this is an extremely exciting time for the disease-modifying therapies project area, since we're funding a range of approaches that utilize unique technologies to identify and discover new antigen specificities. The development of antigen-specific therapies offers the opportunity to target pathogenic cells driving T1D instead of broadly suppressing the immune system. And while therapies that target the broader immune system can be effective for preventing and treating T1D, antigen-specific therapies may serve as another opportunity for a safer and more effective treatment. JDRF currently has multiple shots on goals to lead to the development of antigen-specific T-cell therapies that prevent the destruction of beta cells, and this includes the projects that I showed you today that focus on identifying specific T cells, as well as antigens that drive beta cell destruction. So that wraps up my portion of the talk. And with that, I'll let Jay take it away.
Thank you very much, Cassandra. And hi, everyone. As my colleagues mentioned, my name is Jay Tinklepaugh, and I am also a scientist at JDRF, where I work on disease-modifying therapies with a specific focus on the beta cell. And as my part of today's presentation, I will focus on some of the different approaches that JDRF is pursuing to address the beta cell side of T1D. Specifically, JDRF is supporting projects that are centered on finding new ways to improve beta cell survival and function, identifying proliferative targets to encourage beta cells to proliferate, and we are also supporting projects that are implementing strategies focused on converting other cell types into beta-like cells. Each of these approaches has the potential to help restore insulin production in T1D and contribute to a lasting cure. Now, in keeping with the overall theme of this RIV, several of these beta cell-specific interventions will require targeted approaches so that these promising drugs are delivered only to the beta cell. However, not all of them will need this targeting, and today I hope to demonstrate the varying strategies JDRF is pursuing to have multiple shots on goal in this area. So as we've seen from teplizumab, the exciting results from the BANDIT trial regarding JAK inhibitors and verapamil, which I will discuss in the next slide, non-targeted drugs are incredibly important in T1D, but we will continue to need to evaluate targeted approaches as well. Now, we've mentioned targeted approaches throughout this presentation, but it's worth highlighting that these strategies or targeted drug delivery systems are designed so that any drug or intervention is delivered only to its site of action. For example, as I've shown on the right here, we want to ensure that the drug is only going to the beta cell, the blue cell in this example, and not to any of the others throughout the body. The promise of this approach is that it makes everything safer and decreases the amount of drug needed by being more efficient. This approach has been used successfully in the oncology setting for several years and has improved the efficacy of chemotherapeutic and radiologic treatments while reducing the terrible side effects of those treatments. We want to leverage those advancements made in that field for T1D. So in order to accomplish this goal, there are a number of different drug delivery systems being looked at by JDRF, including using beta cell specific proteins as a form of drug shuttle, nanoparticles loaded up with regenerative therapies targeted to the islets, and even viruses to selectively reprogram cells. Today, I'll be highlighting how these targeted and non-targeted approaches fit within JDRF's overall strategy to impact the beta cell in T1D. One of the first lines of intervention that we can employ in type 1 diabetes is to simply improve the survival and function of beta cells in response to stress and the attacking immune system. Now, just to remind everyone, not every beta cell dies due to the immune system in T1D, and when beta cells are stressed, they can begin to lose function and ultimately die without direct intervention from the immune system. This is a compounding problem in T1D where we know the immune system is also causing widespread damage to beta cells as the disease progresses. So one approach is to systemically deliver an anti-stress agent that provides a benefit to the beta cell. And these anti-stress agents probably don't need to be targeted because of their relatively low safety profile. So previous JDRF research has identified several different pathways in drugs that can keep beta cells alive and functional by making them more resilient to the immune attack. An example of a drug that does this is verapamil. Verapamil is a widely used drug, primarily used to treat high blood pressure, but in mice with diabetes, it was shown to reduce beta cell death, which you can see in the figure. Diabetic mice that were not given verapamil had more beta cell death and thus significantly less insulin production than those that received the drug. So just to highlight it on the right here, mice without verapamil had no protection, whereas those who received verapamil were protected. Now, JDRF has continued to support research on verapamil, and it has been assessed in several small clinical trials over the past couple of years. Currently, we are awaiting the results of the first large-scale trial incorporating verapamil in an early onset T1D population, and we hope to have the results later this year. The second approach is focused on finding ways to get the beta cell that have survived the immune attack and the stress to proliferate or multiply. The goal of this would be to replace the beta cells that were lost by encouraging the survivors to multiply and restore their overall number, as I've shown in the cartoon below. For many years, beta cells were considered part of a small subset of cells within the body that are considered non-proliferative. However, thanks to JDRF-funded research, there have been several exciting discoveries in this area over the years, and we now know how to make beta cells proliferate in vivo. Unfortunately, there are still concerns that these proliferative strategies are not beta cell specific, and they might impact other parts of the body. And this is a huge problem because proliferating cells in the wrong parts of the body could create some major safety concerns. So with proliferative approaches, we will probably need targeted approaches unless we can continue to work on identifying a drug that impacts the beta cell exclusively. And finally, the third approach we are taking involves the conversion of one type of cell into something that much more closely resembles a beta cell. Research has shown that as type 1 diabetes and type 2 diabetes progress, other cells in the pancreas try and change their metabolic processes to help out and supply insulin to the body. Unfortunately, not enough cells are able to change to make up the difference, and people with T1D still need to take exogenous insulin. We are currently researching ways to use gene therapy or chemical interventions to deliver signals to other cells, both inside the pancreas and outside it, to force them to become more like beta cells. This means they can respond to changes in glucose within the body and produce insulin. The hope is that this approach will serve to alleviate the need for exogenous insulin in people who have long-standing T1D. However, as you might expect, we will need to be very careful with this approach and will need highly controlled delivery methods and on-off switches for these converted cells to ensure they are implemented safely. And so any potential cell conversion therapy will almost certainly need a targeted component. Now, the first scientific project I would like to discuss today comes to us from Dr. Assam El-Osta at Monash University in Australia, and it is focused on creating functional beta cell mass. Dr. El-Osta was awarded an innovative award to examine whether a subtype of human pancreatic cells called ductal cells can be used to drive beta cell regeneration. These types of innovative awards are given to projects that are very risky, but have the potential to drive significant advances in the field. Now, as part of this award and part of his earlier research, Dr. El-Osta has shown that the enzyme EZH2 plays a significant role in beta cell development. This work is examining whether chemical inhibition of the EZH2 enzyme can drive the conversion of these so-called pancreatic ductal cells or other pancreatic progenitor cells into insulin-secreting beta-like cells. And so far, the results look promising. In just the 1 year of the award so far, Dr. El-Osta and his team have shown that this strategy was able to restore insulin in the pancreas from a type 1 diabetic donor. And the results of this work were published last year. So while there's still a long way to go with this work and this strategy, there has been some exciting progress in the last year. The second project I would like to discuss comes to us from the company Neurodon and Dr. Russell Dahl, and it is focused on preserving functional beta cell mass. This award is part of JDRF's Industry Development Partnership Program, and through it, Dr. Dahl and his team at Neurodon are designing new compounds designed to prevent beta cell stress. These compounds are specifically designed to help mitigate metabolic dysfunction within the beta cell by activating a specific enzyme called SERCA2, in the hope that this will help them to function properly and contribute to better overall survival. Now, this project also highlights sort of a key role JDRF plays in the research development pipeline, as previous JDRF funding on beta cell stress to researchers such as Dr. Carmela Evans-Molina and Dr. Dezio Iziric laid the foundation for these advancements, and JDRF is sort of uniquely positioned to drive progress from academia to industry and build these types of partnerships that will be key to finding a cure. The next project I'd like to highlight is more of a progress update on the exciting work Dr. Paolo Serafini is doing to develop new ways to increase functional beta cell mass in T1D. So Dr. Serafini's work is focused on using nucleic acid aptamers made of either DNA or RNA to deliver drugs specifically to beta cells. What makes these short sequences of DNA or RNA so special is that they can fold into a shape that will only fit a very specific target. The analogy I've used to explain this in the past is to think of them as the only key to a lock. The folding pattern allows the aptamer to bind tightly to its target so we can deliver a drug with very high specificity. And if you look at the cartoon on the bottom of this slide, you can see a rough interpretation of this folding behavior on the right, and the remainder of the platform can be loaded up with pretty much any drug imaginable. Now, throughout the last year, Dr. Serafini and his team have made exciting progress on this platform and have generated additional evidence that these aptamers can deliver a therapeutic specifically to the beta cell, leading to a high-impact publication. These aptamers are also continuing to be evaluated and examined for their ability to deliver even more therapeutics, and are prime candidates to be integrated with additional targeted drug delivery strategies focused on gene modification and immunotherapies. Now, finally, I'd like to provide two quick updates on other exciting projects JDRF is spearheading. First is from the ongoing collaboration between Mike Cassandra and Ionis. This collaboration was really spearheaded by JDRF, and the central goal of this project has been to design a way to deliver a proliferative agent selectively to the human beta cell. Now, currently, Dr. Sander is transitioning to a new position to direct the Max Delbruck Center for Molecular Medicine in Germany. However, the project is still ongoing, and the partnership is continuing to evaluate these proliferative agents in humanized mouse models of T1D. And second, the partnership between Boehringer Ingelheim and JDRF is continuing to support research focused on identifying novel ways to increase beta cell mass and function in T1D. So to summarize, currently targeted drug delivery approaches are predominantly in the proof of concept stage in T1D, but we at JDRF and across the field are working to keep them moving forward rapidly. And while I think I've made the case that while not everything will need to be targeted, certain interventions will certainly benefit from it. However, there are still some major barriers to progress. First, the cutting edge technology that is being developed here. There have been tremendous advances made in other indications, such as oncology, but a lot of these things are new to T1D, and so they have to be validated and examined before we can really take advantage of them. And second, there are still a limited number of targets that are specific to the beta cell. Now, how is JDRF addressing these barriers? Well, we recently completed a very successful call for applications, specifically focused on bringing more targeted delivery strategies into the portfolio, and many more projects will be joining the JDRF portfolio and starting in the next year. So looking forward, the future of disease-modifying therapies is extremely promising. We will continue to identify discovery research that focuses on new and expanding areas of immune intervention, continue to develop new combination therapies in T1D that address both immune dysfunction and beta cell function, continue to expand the number of targeted drug delivery approaches we have for both the immune system and beta cells, and really continue to drive discovery research into the clinical setting with the goal of developing a cure for type 1 diabetes. So in order to meet these ambitious objectives, we have launched several exciting funding opportunities in the last few months. First off, we launched a request for applications focused on the realignment of established targets and mechanisms for delaying or reversing T1D. This is based on the clear precedent that other therapeutics from other autoimmune diseases might have a beneficial impact in T1D. As I already mentioned, we launched a RFA on the development of islet-targeted drug delivery strategies in T1D with the goal of identifying new beta cell specific targets and developing the tools that can deliver therapeutics selectively to the islet. And then finally, we've also launched a collaborative advancement of cures for T1D RFA. And the goal of this funding opportunity is the development of research collaborations aimed to construct a multifaceted and coordinated approach to advancing the development of cures for T1D. And with that, I'd just like to thank everyone for joining us and for their time and attention. Hope you enjoyed our overview of disease-modifying therapies at JDRF.